Welcome to the deep dive, everyone. Specifically, all you 7th grade life science students out there, plus your teachers and parents. We're really glad you're here.
Yeah, totally. It's great to have you all listening in.
So, to start us off, I want you to picture something. Imagine you were sitting inside an Alaskan field station. Outside the window, a relentless blizzard is just howling across the tundra.
Dropping the temperature way, way below zero. Right?
Oh, absolutely freezing. But inside, it's quiet, the room is warmly lit, and you are surrounded by these beautiful botanical teal blueprints pinned to the wood-paneled walls.
You've got brass and black microscopes lined up perfectly on the workbenches.
Yeah, everywhere you look, there is evidence of really close observation. Anatomical cross-sections of Arctic animals, field journals just filled with charcoal sketches, specimen cards, diagrams of root systems. This is the Bioforge.
It's essentially a place of evidence, of observation, a lab for figuring out the actual mechanical secrets of the living world.
Exactly. Yeah. And when you look closely at those blueprints, this one fundamental rule of biology starts to emerge. And it's our hook for today in science. Shape is often a clue to purpose.
It really is like the ultimate lens for understanding the natural world.
Yeah.
Because when you stop looking at a plant or an animal as just a static object, and start looking at it as an engineered system.
You realize every curve is there for a reason.
Right. Every texture, every internal cavity, it all has a purpose.
Okay, let's unpack this. Our mission for today's deep dive is exploring Crosscutting Concept 6, structure and function. Basically, figuring out how the physical design of something relates directly to what it does.
From, microscopic cells all the way up to massive Alaskan ecosystems.
But I think we need to establish our shared scientific vocabulary first, just so we're all on the exact same page. Can you define structure for us?
Sure. So structure is just how a system is built. It's the material composition, its shape, its internal anatomy, and how all those pieces are spatially arranged.
And functions is the specific job or role that the structure enables.
Right. Exactly. I mean, think of a classic piece of human engineering, like a suspension bridge. The structure is the thick steel cables and the reinforced concrete pillars.
And the specific angles they connect at.
Yeah, the geometry is huge. The function of that structure is to distribute tension and compression so heavy traffic can safely cross a river. The steel doesn't just look cool, its arrangement actively performs a mechanical task.
Which means, if you try to build that exact same bridge structure out of wet cardboard.
Oh, the function completely fails.
Right, because the material properties matter just as much as the shape. And humans have observed this in nature for a long time. I mean, historically, how did scientists move from just looking at things to actually understanding them?
Well, early comparative anatomists would look at these skeletal structures of different animals and notice these striking patterns. If you take the bones in a human arm, the flipper of a whale, the wing of a bat, and, say, the front leg of a dog.
You're looking at homologous structures.
Yes, meaning they share the same underlying skeletal blueprint. Even though a whale is navigating deep ocean currents and a bat is flying around at night, the basic anatomical arrangement of a humerus, radius, ulna, and digits is remarkably similar.
Natural selection just kind of stretched or shortened those specific bones over millions of years to adapt them.
Right, to entirely different functions.
But wait, can't appearances be deceiving? Like, if we just look at the shape of something, couldn't an anatomist easily guess its job completely wrong?
Oh, they absolutely could. And the history of science is actually filled with those exact mistakes. Visual observation is really only the first step. To truly understand function, scientists have to test the structure.
Which brings up a Bioforge favorite, the sea otter. What did early naturalists get wrong about them?
Well, they totally misunderstood their teeth. Sea otter molars have a very specific shape known as bunodont. A buno means mound or hill, and odont means tooth.
So they aren't sharp like a wolf's.
No, not jagged for slicing meat, and they aren't flat with rough ridges for grinding grass like a cow has. They're these wide, heavily rounded, bulbous molars.
So what did the early scientists think they were for?
They looked at that flat, bumpy structure and assumed it functioned like a mortar and pestle, for grinding up tough marine algae and kelp.
I mean, that sounds like a perfectly reasonable guess based on the shape alone.
Reasonable, yeah, but entirely incorrect. This raises an important question about how we verify our observations when later biologists actually gathered empirical evidence.
Like checking stomach contents.
Exactly, checking stomach contents and conducting actual mechanical crushing tests on the teeth. They discovered the true function. Sea otters don't eat kelp. They eat heavily armored invertebrates like sea urchins and thick-shelled clams.
But really, so if an otter had sharp pointed teeth, what would happen?
The intense pressure required to bite through a clamshell would just snap a sharp tooth right in half. But that rounded, bunodont structure mechanically distributes the extreme vertical force across a wider surface area.
Oh, wow. So it lets them fracture a calcified shell without shattering their own skull.
Exactly. So the initial guess was logical, but the empirical evidence told the real story. Scientists study parts closely, but they really have to prove what those parts do.
Evidence over guesses. And our scientific explanations improve massively when new tools reveal structures we couldn't see before, like William Harvey in the 17th century.
Oh, a fantastic historical milestone. Before Harvey, the function of the heart was heavily debated. Some physicians thought it was like a furnace that literally heated the blood.
Or that blood just sloshed back and forth randomly.
Right. But Harvey didn't just guess. He applied a mechanistic lens. He dissected hearts and observed the structure of the valves between the chambers.
And a valve only opens in one direction.
Yes. He demonstrated that the physical flaps of tissue are structured to fold back when blood pushes forward, but they billow out and lock together if blood tries to flow backward.
Proving that its function is to pump blood in a continuous one-way circuit. Here's where it gets really interesting, because once we got microscopes, we could see totally new invisible structures. So let's zoom all the way in. If we put a slide under one of these brass microscopes in the Bioforge and look at a cell, how are structure and function operating at that microscopic scale?
Well, at the cellular level, the physical boundaries are incredible. Take the cell membrane. Its structure is a phospholipid bilayer. Picture a double layer of specialized fat molecules.
Okay, I'm picturing it.
The outer heads of these molecules are attracted to water, but the inner tails repel water.
Wait, if the cell is mostly water inside and lives in a watery environment outside, but the membrane has a water-repelling layer, how does it get nutrients in? It sounds like a sealed Ziploc bag.
That's the genius of its structure. Because the tails repel water, random molecules can't just drift across. It's a highly effective barrier. But embedded in that fat layer are specialized protein channels.
Sort of like a high-tech security gate at our field station.
That's a perfect analogy. These proteins are structured like microscopic tunnels with highly specific shapes. Only a molecule that perfectly fits the shape of the tunnel can get in. The function is to act as a selectively permeable boundary.
It strictly controls the internal environment. That is so cool. And inside that boundary, you have organelles.
Right. Specialized substructures with distinct jobs. In a plant cell, you have a rigid cell wall made of tough cellulose. That structure allows cells to stack like bricks, providing support so the plant can grow toward the sun.
And they have chloroplasts for photosynthesis.
Yes. Now, animal cells lack those rigid walls, but they have a different structural marvel, the mitochondria, the powerhouse of the cell. If you look at one under an electron microscope, the inner membrane is folded back and forth on itself countless times.
I always wondered about those folds. Why wouldn't it just be a smooth, round balloon shape?
Surface area.
Yeah.
The chemical reactions that synthesize ATP, the energy currency for the animal, take place directly on that inner membrane.
Oh, I get it. Like trying to fit a massive sheet of paper into a tiny matchbox. If it's flat, it won't fit. But if you crumple it up, creating hundreds of folds.
You pack a massive amount of surface area into a tiny volume. The folded structure maximizes ATP production.
Okay, so that's the micro scale, but how does that scale up?
Well, cells organize into tissues. Tissues form organs. Organs form body systems like the nervous or circulatory systems, and they constantly interact.
Let's picture that out in the Alaskan wilderness. If an animal, like a caribou, is running from a predator, what's happening?
The nervous system detects that the muscle tissues are burning ATP and need oxygen. It signals the respiratory system to breathe faster, and the circulatory system pumps blood harder. The blood vessels literally dilate, widening their structure to deliver that oxygen.
All those microscopic systems working together so the caribou can survive in the macro world, which brings us out into the extreme environments of Alaska.
Where the margin for error is incredibly thin, take the Pacific salmon. Their primary structural adaptation for swimming upstream is their fusiform body profile. They are essentially shaped like a living torpedo.
Which minimizes hydrodynamic drag.
Right, the water slips seamlessly over them, functioning to conserve energy against freezing currents. But they also have this small, fleshy nub on their back called the adipose fin.
Didn't people used to think that fin didn't do anything?
Yeah, they assumed it was a useless leftover. but it's packed with nerves. It functions as a highly sensitive flow sensor. When a salmon navigates rocky rapids, the fin physically bends in response to turbulent water, sending neural feedback so the fish can adjust its swimming path.
Wait, so different parts of the exact same animal are doing entirely different jobs to achieve one overarching goal.
Exactly. Survival and reproduction. We see this with Alaskan bears, too. A grizzly bear has long, blunt, gently curved claws and a massive shoulder muscle hump.
Built kind of like a heavy shovel, right?
Yes, a structure optimized for digging up roots and excavating winter dens. Whereas a black bear lacks the hump and has shorter, tightly curved, needle-like claws.
Perfect for gripping bark and climbing trees. Form dictates behavior. But what about the cold? That's the biggest constraint in Alaska. Sea otters don't have thick blubber.
No, they rely entirely on the structure of their fur. They have up to 1 million hairs per square inch. The fibers have tiny interlocking scales that trap a permanent layer of insulating air. The freezing water never actually touches the otter's skin.
Incredible. And out on the tundra, the muskox has to manage heat loss. They have a really stocky, barrel-shaped body.
That geometry minimizes their surface area to volume ratio to prevent heat loss. Plus, they have a dual layer coat, long guard hairs on the outside, and an ultrafine underwool called quiviate.
And quiviate is what, 8 times warmer than sheep's wool?
Because it lacks the microscopic scales of normal wool, it packs tightly, trapping millions of tiny pockets of dead air. It's the ultimate structural insulator.
Now contrast that with a moose. A moose has incredibly long legs that let it navigate 36 inches of deep snow. And their massive body is great for retaining heat in the winter.
But that same structure becomes a huge constraint in the summer. A massive body traps too much heat, causing severe thermal stress. They are physically forced to alter their behavior, like standing chest-deep in cold lakes just to survive an 80-degree day.
Wow. Even the frogs have structural defenses. The freeze-tolerant wood frog pumps massive amounts of glucose into its cells before winter.
The glucose functions as a biological antifreeze. It prevents the water inside the cells from forming sharp ice crystals that would physically rupture the cell membrane. The frog can survive being 60% frozen solid.
And tundra lichen do something similar, right? Their thick outer walls are so elastic they just stretch during freeze-thaw cycles without breaking.
Exactly. Even plants engineer their way around the cold. Out on the exposed tundra, dwarf shrubs, like the dwarf willow, grow prostrate, meaning very low to the ground.
It stays out of the wind.
Yeah, it stays within the warmer boundary layer of the earth. And spruce cones feature tough, woody scales that close tightly in cold, damp conditions to protect the seeds.
And flex open in warm weather to let the wind carry them away. It's amazing. And all of this connects right back to humans. Because Alaska native communities have thrived here for thousands of years by observing these exact structures and functions.
If we connect this to the bigger picture, traditional ecological knowledge, or tech, is a sophisticated, highly empirical science. It's based on millennia of quiet, systematic observation of animals and environments.
Form perfectly supports function in indigenous tools, like the ulu or uluak. It's a crescent-shaped blade, and the handle is hafted directly over the center of the cutting edge.
That structure fundamentally alters the biomechanics. It multiplies your downward force and completely eliminates wrist torque, so you can process fish and hides with minimal physical span.
Then you have snowshoes, or tegluk, a broad birch wood frame with moose skin webbing.
It distributes your weight over a wider surface area. It decreases the pounds per square inch, so the wearer doesn't just sink into deep powder.
And indigenous fish traps and weirs. They use funnel shapes designed with specific angles relative to the salmon's body dimensions.
Right, a salmon has a wide turning radius. The trap is structured so the fish can swim in, but physically it cannot pivot to escape.
And they tied those traps together using spruce root lashing. Total mastery of material science. The specific lashing patterns absorbed the force of the rushing river. It's so important to note that this cultural knowledge is profoundly place-based.
It's practical, relational, and deeply connected to a responsibility to steward the environment for future generations.
So what does this all mean for you, the listener, sitting in your 7th grade classroom? How do you apply this?
Well, in the Bioforge lab, we use a Harvard Project Zero thinking routine called parts, purposes, and complexities.
How does that work?
First, you observe and list the parts of a system. Like, what is the shape of a bird's beak? Second, determine the purpose or the function of each part. Does it crack seeds or extract nectar? And third, explore the complexities. How do the parts work together? And what happens if one goes missing?
What's fascinating here is that this routine proves science is an active process of figuring out. You aren't just memorizing facts from a textbook.
You're decoding the world.
From the folded membranes of a mitochondria to the crescent blade of an ulu, the physical arrangement of materials dictates what a system can achieve.
It's a universal law.
Which brings us to the final rule of today's deep dive. Form gives scientists clues, but evidence explains function.
And I want to leave you with one final provocative thought to mull over. If natural selection shapes biological structures over millions of years, and human engineers use biomimicry to design tools based on those principles, how might the rapidly changing climate in Alaska force both animals and human engineers to radically redesign their structures in the next few decades?
Will there be a point where a previously perfect structure becomes a fatal flaw?
It's a huge question.
It really is. So here is your final challenge for today. Look around your own room, or look out into your backyard right now and finish this sentence. One structure I can observe and connect to a function is